I'm very excited to share with you an overview of a study where we employed a high-throughput proximal tubule on a chip platform for nephrotoxicity detection. And I think at the very beginning, it's really important to note what a great collaborative effort this was. We had colleagues participating from MEMETIS, as well as from Radboud University, the University of Applied Sciences, Northwestern Switzerland. We actually heard from Laura earlier today. And we also participated together with Utrecht Institute for Pharmaceutical Sciences. I'd also like to point out that we collaborated closely with 3 different pharmaceutical companies. So that's GSK, Roche, and Pfizer, who actually provided blinded compounds for the study. So let's jump in. So drug-induced kidney injury is a dose-limiting factor in pharmacotherapy, and it's often associated with kidney failure in many patients. We know specifically that especially the renal proximal tubules are susceptible to drug-induced kidney injury due to their active role in the secretion of xenobiotics into the urine. Now, currently in preclinical drug development, only about 2% of drugs actually fail due to nephrotoxicity. And this ends up leading to 75% of clinical nephrotoxicity that's not detected in that preclinical phase. So really taken together, these data suggest that in order to prevent drug-induced kidney injury, to save patient lives, we need to detect it earlier on in the drug development pipeline using complex in vitro models that are robust and reproducible. However, many of those out there have a number of limitations, including the complexity of the model, the fact that they're low throughput, maybe they only have a single chip or they're connected to pumps. They often lack the apical and basolateral accessibility of the proximal tubule, and they finally lack the cell ECM interaction, perhaps due to the presence of a semi-permeable membrane. Now, there is a platform out there that addresses many of these limitations, and that's the MEMETIS organoplate. So perhaps you've already heard about the organoplate previously during the meeting, but I'd like to just give you a bit of an overview. So here we see the organoplate bottom is made up of microscope quality glass. It's situated in a microtiter plate, and our chips are actually embedded within that glass, and we can have anywhere from 40 to 96 individual tissue chips within the organoplate, so nice and high throughput. Now, if we look at the top of the plate, it may look a little bit more familiar to you, similar to other 384 well plates you may have seen before. So you can use a pipette or an automated liquid handler to add your cells, extracellular matrix, and media to build your stratified tissues layer by layer within the microfluidic channels. There should be a video here for you, but it's not starting. I wonder if from the back of the room, would you be able to start the video? Excellent, thank you very much. So now this video will just give you a bit of an overview of how the organoplate works. So we're gonna go ahead and zoom in on a single tissue chip. We see we have 3 lanes running in parallel. This is our 3-lane 40. Now there's no physical barriers that are separating the 3 lanes. Instead, we have these short ridges of plastic called phase guides that are capillary pressure barriers. So when you pipette in your extracellular matrix gel, the capillary pressure barriers pin the gel within the channel and prevent spilling over into the perfusion channels. Once solid, we can add cells like proximal tubule cells to that perfusion channel. They adhere to the ECM, and we can begin perfusion by simply placing the plate on a rocker system within the incubator. And the wells of the microtiter plate act as medium reservoirs, such that as the plate rocks back and forth, the media flows through those microfluidic channels. And then it's programmed to rock back in the other direction. And over time, you'll form this 3D proximal tubule structure in that perfusion channel. So there are many flavors of organoplates. I'd say the simplest is first our 2-lane organoplate. So this one has only 2 microfluidic channels in parallel, separated by a single phase guide. And we can have up to 96 chips on this plate. So we use this commonly for our liver model, where we have hepatocytes embedded in the gel and endothelial cells and Kupffer cells against that channel. Now, we already discussed a little bit the organoplate 3-lane, and you'll see that later in the presentation for our proximal tubule model. We've recently developed the organoplate 3-lane 64. So this not only has increased throughput to the 40, but it also is more compatible with high throughput automated liquid handling. And then finally, we have the organoplate graft, which is a bit of a variation on the 3-lane in that the gel channel is open on the top so that you can graft on a spheroid, a tumoroid, or organoid tissue, and perhaps have it vascularized by a bed that's within that ECM. So I hope you can begin to appreciate that the organoplate is one platform for all cultures, and it often addresses the drawbacks of many of the classic laboratory setups. I think particularly when thinking about transwell cultures, we are membrane free. We also have, that nice access to apical and basolateral sides. We can introduce perfusion to the cultures, which we find that fluid shear stress enhances the differentiation of the cell types frequently. The throughput, as we already discussed, and it's quite compatible because of that microtiter plate format with those automated liquid handlers, but also plate readers and high content imagers that you already have in the lab. And there's some really nice examples of some beautiful confocal images you can see on the screen. So we can see through that microscope quality glass, you can easily capture these beautiful images that are also quantifiable. Okay, so now we can dive into the proximal tubule on a chip and the Nephroscreen study that we performed. So to set up our proximal tubule on a chip, we used the organoplate 3-lane and actually used 2 different types of cell lines. So the first was a conditionally immortalized proximal tubule epithelial cell that overexpressed the OAT1, organic anion transporter 1, which is an uptake transporter that plays a role in toxicity in the kidney. And the other was using immortalized RP-TEX from Sigma. And we found when we exposed them, when we added them to that perfusion channel, exposed them to shear stress, we observed some really nice differentiation of the cultures where we observe an apical and basolateral side of those proximal tubules. So to assess drug-induced kidney injury, we used 4 model nephrotoxic drugs, in addition to those 8 blinded compounds that I mentioned earlier that were supplied by pharmaceutical companies. For each drug, we used 2 concentrations and selected 2 time points. And we evaluated a number of different endpoints across multiple laboratories. And we'll see that in the next slide. And I think that really lends to the robustness and the reproducibility of the platform. So here we have a bit of our screening workflow, each for the OAT1 cells and the RP-TEX. And we can see that each rectangle represents the location where that assay was performed. So for example, on the left-hand side, if we look at the OAT1 cells, Mehmet performed all of those that are highlighted in green, while folks at Radboud University, all of those in blue. And then at the University of Applied Sciences, those rectangles in red. So really big collaborative effort. And then in the next coming slides, we're not going to review all of the data. I'll just give you a bit of an overview. So if you are interested in more information, I invite you to visit our publication, which is listed below. So first is the barrier integrity assay. So this is a measure of the tight junction function of our proximal tubules. And it tells us a little bit about the health of the cultures. And we can do this by using a fluorescent dye, like a FITSI dextran dye, and we add that to the perfusion channel. And if our tubes are intact and leak tight, then you'd expect that fluorescent dye to stay within the perfusion channel. And you can see that over here, we have a nice example of a leak-tight tubule, so that fluorescent dye is staying in the top perfusion channel. However, if we've applied a toxicant that's disrupting the cells, disrupting the tight junctions, then you're going to see leaking of the fluorescent dye into the gel channel. And that's what we see in our next fluorescent image. So what we can do is actually quantify the fluorescence intensity in the perfusion channel as well as the gel channel to calculate the apparent permeability. And when the apparent permeability is low, then we actually have a really nice and great intact barrier. However, when the apparent permeability increases, we know we're losing our barrier function. So if we look over on the right-hand side, just a review of our results in the RP-Tech cells, we have one graph on the left that features results from the 24-hour time point and then 48-hour time point to the right of that. And on the y-axis, we have PAP value and then all of our blinded compounds across the bottom on the x-axis. So I just want to point out one example to give you an idea of how to interpret the graphs. So if we look at the very first gray bar, we see that we have a nice and low PAP value, right? So that means that we have great intact barriers. However, when we apply G1, so that's our first blinded compound, we see a dramatic increase in that PAP value, suggesting that we're losing barrier integrity. And even at the higher dose, you notice that you don't even see a bar there. And that's because when you completely lose the barrier function, you're actually unable to calculate that PAP value. So when you look even at the 48-hour time point, both G1, the low concentration of that blinded compound, and the high concentration of that blinded compound, we've completely lost the barrier function, suggesting that the longer time points induce more toxicity. But overall, the blinded compounds, they did show different effects on the barrier functions. So one of the other assays we employed was the WST8 assay. So it's an indirect measure of viability. And same here, we actually found that there was just varying effects on the different blinded compounds. This was particularly in our OAT1 overexpressing cells. And there was an overall trend for the 48-hour time point to have a greater effect than the 24-hour time point. We also evaluated LDH. So this is a cytotoxicity measure. We know that when cells are under stress, they'll release LDH into the media. So we sampled the media from our blinded compounds, and it was a similar effect observed here. So just different effects for those blinded compounds that was measured by the release of LDH. So this table I just wanted to share with you so that you could take away the fact that there were a lot of data points collected for this study. So over 2,000, I think we had over 2,600 actually data points that were collected for this study. So I would say it's pretty medium to high throughput evaluation. I definitely don't expect you to be able to read and interpret everything that's going on here. But I hope in the next slide, I can give you some of the insights that we took away from this study. So what we found was that most of the blinded compounds, that was 7 out of 8 of them, resulted in toxicity by at least one of the parameters or in some of the drug transporter interactions. So this is 87% of the time we detected nephrotoxicity, and this is threefold better than our current preclinical tests. We observed a decrease in barrier integrity, and that's often coupled with a decrease in cell viability, as well as an increase in LDH release. So we can say that the loss of the barrier integrity is directly associated with the cytotoxicity. We also found that prediction of nephrotoxic liability was highest when the results of multiple assays were combined. So cell viability, LDH release, as well as microRNA release. And then as we kind of were seeing throughout the slides, exposure for 24 and 48 hours provided sufficient, were sufficient to detect drug-induced nephrotoxicity in this case for evaluating acute toxicity. But I do think it's important to note that the platform can also be implemented for maybe compounds that require a longer or subchronic exposure. So I hope you can see that our proximal tubule on a chip is really a robust and transferable platform, which is going to be key for that broad implementation into industry. We're making use of some well-characterized proximal tubule epithelial cell lines, and we like that they're a potential alternative for the use of animal studies. And then finally, I really hope that I've convinced you that the Nephroscreen, this high throughput proximal tubule on a chip, is a reliable, standardized, automatable system that was really useful in identifying nephrotoxicants and provides insights in some of the mechanisms of toxicity. So actually, I want to give you a little bit of a teaser and let you know, if we can go back, there we go, how we're building more complexity into our proximal tubule on a chip and modeling ischemia reperfusion injury. So in this case, we've included not only our RP-Tech cells, but also our HUVEC cells in the bottom perfusion channel. And we were able to mimic ischemia reperfusion by exposing the cultures to low oxygen, keeping them under static conditions and having them with low glucose. So we did that for 24 hours. And then the reperfusion part came where we basically just reintroduced them to normal conditions. So perfusion, higher oxygen, normal glucose. And what we observed is that in that ischemia reperfusion condition, we actually disrupted those proximal tubules. And we can see that by the fact that we lost, our proximal tubule cells. We can see some DNA staining here. We also see some caspase 3-7 staining, suggesting that there's some apoptosis going on. But I think what was really cool is that when we applied adenosine to the culture to try and prevent injury, we observed that the adenosine actually rescued this injury. So lastly, kind of building even further on that proximal tubule, we know that the proximal tubule compartment is not the only part of the kidney. The glomerulus is also an important part of the nephron. So we've collaborated closely with Dr. Laura Perrin at Children's Hospital NLA, who's developed a glomerulus on a chip in the organoplate 3 lane. And we plan to transfer the media from the glomerulus on a chip to the proximal tubule on a chip, as similar as we can right now to be what's going on in vivo. And in our preliminary studies, we used membranous nephropathy as a way to first characterize the system. So membranous nephropathy is a rare kidney disease. It's an autoimmune kidney disease where patients often exhibit loss of kidney function by seeing albumin in the urine. And these patients, they frequently have a lot of circulating anti-PLA2R antibodies, which are specific to the podocytes in the glomerulus. And so for us, and Laura has done this already, where she's been able to, in the glomerulus, model membranous nephropathy by applying patient serum, and we observe a loss of glomerular function. But our question was, how does this loss of glomerular function actually impact the proximal tubules? So if we look on the right-hand side, after treating the glomerulus and then applying it to the proximal tubule, we first confirmed that we have a loss of glomerular function when we applied the patient serum to the glomerulus. However, after applying the filtrate from the glomerulus to the proximal tubule chip, we see also that the proximal tubules become damaged because we see an increase in KIM1 secretion. So with that, I really hope I've convinced you that Mimetis develops human tissue models for better therapies, that we're unlocking novel biology for better and faster assays to have an impact on therapy development. With that, I would really like to acknowledge all of my colleagues at Mimetis, as well as all of the folks that participated in the Nephroscreen. And I'd like to thank all of you for your attention, and I'm happy to take any questions.
Thanks, Chris. I think we have time for one or two questions.
Thank you for your talk. This is Ramin from Kyoto University. I was going to ask if you have investigated formation of bilayer epithelial and endothelial until you showed the last picture. And I was wondering if you could make this bilayer more intimate, like culturing HUVEC right in contact with RPTEX. That's question one. Question two, the difference between your OAT1 representing cells and the immortalized one. How much is the difference in amount of OAT1 actually?
Good questions. So I think there's a couple ways we can get those proximal tubules and the HUVECs closer together. So we can, and I didn't mention this, for our glomerulus model, we actually culture in one channel the podocytes and the endothelium. So you can kind of build a tube within a tube so that the cells are directly layered on top of each other. So I do think that that could be a potential option. Another one could be making that gel channel, perhaps a little bit smaller. Yeah.
Okay.
And then your second question, would you mind repeating it? I think you were asking the difference between the OAT1 overexpressing cells and the RPTEX cells.
Yes, yes.
So it's primarily going to be the fact that OAT1 is very highly expressed in those overexpressing cells. I do believe they also have a very nice expression of PGP and MRP2. RPTEX have them as well, but I'm not sure if you noticed in the table, actually, when we applied our known compounds, we had more of an effect in the OAT1 overexpressing cells than in the RPTEX cells. And I suspect it's due to the expression of the transporters, higher expression of really, most of the transporters.
Very well. Thank you very much.
Yep.
I have one quick question if I'm allowed. When you did this round, the study, you had two labs checking it, which is a good idea. Did any of the pharma partners actually set this up or try it?
To my knowledge, they did not, no. Yeah, but that would be interesting. Will they? I don't think they're planning.
And I mean, did you get any information about the 8 compounds? Because they're obviously positive nephrotoxins. Are they clinically positive, only positive in animals?
So I think a little bit of both. And actually there were some in there that weren't nephrotoxic. There was at least 1 or 2, yeah.
And you flagged them up as positive?
So all but 1.